Good afternoon, everybody. And today I'm going to talk about the first allogenic mesenchymal stromal cell product, Stempeutics, approved for marketing by the Indian FDA for critical limb ischemia management. About Stempeutics, which is a biotech company which is focusing on development of bone marrow-derived MSC product from healthy adult volunteers. We are founded in 2006 by the Manipal Group, which is headquartered in Bangalore, India. Till now, we have filed 60 for around 60 plus patents and published around 75 peer-reviewed international publications. And apart from this indication of these two indications, budget disease and atherosclerotic peripheral artery disease, for which actually we have got the marketing authorization, we are conducting phase 3 clinical trial in two indications, osteoarthritis and diabetic foot ulcer, which are in advanced stage of phase 3. And apart from that, we are conducting a phase 2 clinical trial in perineal fistula due to Crohn's disease. About this Stempeutics product, it is an on-demand, cryopreserved, allogenic regenerative medicine product targeting the major unmet medical need. And this is the first pooled stem cell product for which we are given awarded the ATMP designation and ODD designation by EMA in 2015 for the indication CLI due to Berger disease. We have awarded 18 international patents, which for the pooling technology, which includes in countries like US, France, UK, Germany, amongst others. And apart from the other trials, we are also going to initiate a COVID-19 ARDS study. So what is the active Stempeutics, which is a pooled product, and the process is a highly scalable process which has got one-time collection. And with this one-time collection, we have got a working cell bank sufficient to produce 1 million Stempeutics cell units. So this solves a major manufacturing issue in the regenerative medicine field. What we do is that we screen actually multiple donors as per our internal SOP, and we do also extensive screening of the infectious disease markers, and then from there we select 3 healthy donors. We aspirate around 60 to 100 ml of the bone marrow. From there we isolate the MNCs and we expand them to bone marrow MSCs till passage 1. So till passage 1, we have got the individual master cell banks of donor 1, donor 2, donor 3, which have been cryopreserved. Thereafter, we take 1 vial each of donor 1, donor 2, and donor 3, pool them, and then we expand them further to passage 3, and this becomes a pooled working cell bank, which is again been cryopreserved. Then whenever we require the final product, we expand the working cell bank to the final product in passage 5. At each stage of MCB, WCB, and IMP, we do extensive testing for endotoxin, mycoplasma, fungi, and amongst others. We characterize them for the positive and plus the negative markers by flow cytometry. And apart from that, we do the karyotyping and the differentiation assays, and also we do the DNA ploiding. So what is the advantages of this pooling technology? It also gives us a broader cytokine growth factor array as compared to that of single donor. We have got a large product doses versus a single donor. And the most important thing is it reduces the variability amongst the donor as it balances the individual variability in terms of proliferation, low immunogenicity, and stable or consistent immunosuppressive activity. And it also gives a stable secretion of growth factor and wider array of growth factors, and especially the stable secretion of angiogenic factors. And lastly, we have got a lower cost of production. So about this CLI can be due to peripheral artery disease, due to atherosclerosis, or due to Berger disease. Atherosclerotic Ph.D. is basically because of deposition of atherosclerotic plaque or actually that are fatty plaques. The risk factors, underlying risk factors include smoking, diabetes, obesity, cardiovascular problems, and hypertension. Berger disease is because of deposition of inflammatory thrombi in the vessels, maybe because of excessive smoking. Ultimately, this patient actually manifests with rest pain, ulcers, or gangrene. But there are certain gaps in the treatment option as of now because of a Berger disease. The symptom they manifest in a very advanced stage of disease. So by stopping smoking will not stop the progress of the disease. And because of the diffuse and distal nature of the disease, because the small vessels are affected in the distal limbs, so the revascularization surgery also may not be effective in these patients. Ultimately, the patients progress to limb amputation. For 25% of the patients, progress to limb amputation over the span of 5 years, and there are still no drugs approved for Berger disease. The only symptomatic treatment is being given. Similarly, for atherosclerotic PAD, the current standard of care includes medication, endovascular surgery, angioplasty, or atherectomy. and there is a very high risk of leg amputation and death. The fate of patients when actually they present the primary treatment, only 50% of the patients are eligible for revascularization and 25% of the patient go for amputation. But if you see the patient after 1 year, only for a CLI resolved in only 25% of the patient, and 25% of the patient, there is a very increased risk of mortality, and of course, the 30% of the patient, they have been amputated. Most important thing is only 15% of the amputated patient, they lead a normal life. Your stem cell, because of the property of anti-inflammatory, ability to promote the neovascularization, and promote healing of non-healing ischemic ulcers, may be useful for this major unmet medical need. The business opportunity for these two indications are huge because the Berger disease in most of the countries, they do fall into to the ODD for the designation, and the CLI pad also because of the increased incidence of diabetes, the number of cases are increasing across the globe. If you see the global business opportunity, if we take both the indications, comes to around $5.5 to $6 billion, and it is gradually increasing on. So we have conducted an preclinical efficacy study in a biopsy nude mice, where we have where we done the ligation of the femoral vessels, and we injected 5 million cells at the site of the ligation, and we followed the animals for a total duration of 28 days, and there were 3 different groups: stem cell group, the vehicle control group, and the sham control. What we have seen is that in the stem cell group, there is only evidence of this toe necrosis at 28 days follow-up. Vehicle control group, there is evidence of total loss of limb, and sham control everything within normal limits. And also we looked at the foot necrosis. What we saw in the sham control, which is the blue line, there's 100% protection against the foot necrosis. In case of vehicle control, all the animals had foot necrosis within 7 days. Once we inject the stem cell group, the 70 to 80% of the animals had protection against foot necrosis. So we also looked at this necrosis score, the ambulatory score, a span of 28 days. This orange bar is the vehicle control, green bar is the stem cell group. What we saw at different time points of the follow-up, this comparing the stem cell group with the vehicle control was found to be statistically significant. So with this, we can safely conclude the intramuscular administration of stem cell ameliorates the neck necrosis and improves the limb salvage in hind limb ischemia model. So what is the mechanism of action? Ischemia in patients, they do cause inflammation, loss of the muscle tissue, which can be seen histopathologically also, with the increased evidence of necrosis and increased inflammatory cell infiltrate. And stem cell, because of its properties, it does cause evidence of angiogenesis, it causes evidence of immunomodulation and reduction in inflammation, and evidence of this muscle regeneration because of increased secretion of anti-inflammatory cytokines and also the angiogenic cytokines. Outcomes to the patient are relief of rest pain, healing of the non-healing ulcers, increase in APPI, which is an indirect evidence of neoangiogenesis, and increased collaterals as seen by MRI. The label of the drug which is approved by Indian FDA is CLI due to Berger disease or atherosclerotic peripheral artery disease in the Rutherford grade 5 or 6, not eligible for or have failed traditional revascularization treatment with rest pain ulcers in the affected limb. The contraindications are with a known hypersensitivity to DMSO HSA or to known sensitivity to porcine or bovine product, and it has been recommended to measure the oxygen saturation by pulse oximetry during infusion and also 2 hours after the infusion. The stem cell has been granted the marketing authorization by Indian FDA. So how do we inject the cells? We make a grid 10 centimeter by 6 centimeter in the calf muscles. We give multiple injections, 40 to 60 injections in the dose of 2 million cells per kg body weight. Patient being under IV sedation or short general anesthesia. And if the patient is present with ulcer, we give 8 million cells peri-ulcer injections also. And apart from that, we give a pre-medication of hydrocortisone and phenylenediamine by intravenous injection. We have conducted a phase 2 clinical trial in the Berger disease in 90 patients. It was a dose-finding study, 1 million cells per kg body weight and 2 million cells per kg body weight, 36 patients each, and the control arm, 18 patients. And the 6-month data is presented here. What we see is that this orange line, which is our 2 million cells, a blue line, which is our control arm, for which the relief of rest pain was the primary endpoint. And what we see at the 6 months follow-up, the rate of relief of rest pain was compared to control arm in 2 million cells per kg body weight was statistically significant. Similarly, the ulcer healing, which was again the primary endpoint, in this what we saw is for the orange line, the rate of ulcer healing and 2 million cells compared to control arm was also found to be statistically significant. And apart from this, we also looked at the ABPI and what we saw for the dose of 2 million cells per kg body weight, ABPI increased from 0.47 to 0.66 at 6 months follow-up as compared to control arm, which remains static at 0.68, came to 0.67. And this difference between 2 million cells and control arm was found to be significant. So phase 2 trial obtained for Berger disease demonstrates statistically significant superiority over standard of care for dose of 2 million cells per kg body weight. Then we proceeded to conduct our phase 4 study on Berger disease, and in 50 patients, what we have seen, the same parameters, rest pain, ankle systolic pressure, ABPI, and ulcer healing, they were all statistically significant when compared to the baseline. For example, for ABPI, the ABPI increased from 0.44 the baseline to 0.76. The difference was statistically significant. Any increase of ABPI more than 0.15 in these patients is also clinically beneficial. For ulcer healing, total of 59 ulcers were present at the baseline at 6 months follow-up. Out of this, 40 ulcers completely healed, 12 ulcers improved, and there was 3 ulcers did not improve. So with this data, we went to the Indian FDA to give us a label extension phase 3 study approval for atherosclerotic Ph.D. in 24 patients. We were granted the approval. We conducted the study, and we followed the patient for 6 months duration. And what we saw, the same endpoint, rest pain, ulcer healing, ankle systolic pressure, ABPI, they were all statistically significant when compared to baseline. Here also we had the same data of ABPI increasing from 0.47 to 0.71 at 6 months follow-up, which was statistically significant. When we compare this data of our CLI phase 3 Ph.D. study with a historical control of 18 patients, all of the endpoints of rest pain, ulcer healing, ABPI, ankle systolic pressure, they were all statistically significant when compared to the control arm. For example, in ABPI, the control arm orange line remains same, 0.6 at baseline to 0.6 at 6 months, whereas in cases of the cell arm, decreased from 0.5 to 0.7, which was statistically significant. Adverse events, there were a total of 184 patients of CLI which were been injected, 156 in the cell arm and 28 in the placebo arm. And the common adverse events were skin ulcer, pyrexia, gangrene toe amputation. All adverse events were related to the progression of the disease, and they were and all the SAEs were not related to the drug as opined by the investigators, ethics committee, and the DSMB. This is the photograph of one patient having ulcer at baseline, which you can see which are completely healed at 6 months follow-up. Apart from rest pain, also in this patient decreased, the ABPI also increased. And another patient having a large ulcer, which also completely healed at 6 months follow-up. And this is a photograph of MRA, where we can show that the 6 months follow-up, the anti-tibial artery has been completely reformed. And another patient, there was evidence of the posterior collaterals in this patient at 6 months follow-up. And this is the CLI Ph.D. phase 3 study. What we have seen is that the ulcer, large ulcer like 7.67 centimeters, 7.28 square centimeters, they completely healed at the 6 months follow-up. So to summarize my talk, total of 184 patients has been injected, in which we have phase 1 study, phase 2 study, phase 4 study for Berger disease, and the label extension phase 3 study in Ph.D. In the phase 1 study, which are 20 patients, the proof of concept study in which ABPI and ankle systolic pressure index, they were statistically significant. Phase 2 study, dose finding study, 2 million cells per kg body weight, all the parameters measured, rest pain, ulcer healing, ABPI, ankle systolic pressure index, total walking distance, quality of life, all were statistically significant. Similarly, phase 4 study and CLI PAD, all the parameters were statistically significant. So safety and efficacy of Stempeutics has been established. 2 million per kg body weight is the most efficacious dose. With this data, the Indian FDA has granted marketing authorization for CLI due to Berger disease and CLI due to atherosclerotic Ph.D. And this drug will be available from Q4 in 2020 in India for the active marketing. And now we are looking for partners for expansion to US, Europe, Japan, and conduct of a global clinical trial for CLI due to Berger disease. Thank you for your attention.